We tabulated site and systems false run rejection rates, and that was eye-opening too. For some of the well-performing, very high sigma methods, we went to 1,3S rules and did a lot less repeat of quality control. We also, as we went through this, took a close look at our peer comparisons, our CAP proficiency, to make sure there was no quality degradation as we went through the process of changing to Sigma for our basic quality control. And I must say our CAP comparative proficiency testing results are well above average. There was no degradation. So the worst case scenario would be we'd opened up Westgard rules too wide and we suffered degradation. That did not happen. And we're in the process of expanding the Sigma approach to hematology and coag. I'll be showing you largely chemistry data, but Sigma is applicable to many different areas of the laboratory. Dr. Harrison, who had to leave a little bit early, gave a poster showing how Sigma could be used with Stagos in coagulation testing in our enterprise. Again, using the same standardizing, measuring, monitoring, and evaluating. So this is just a diagram that shows you the connectivity. The wide area network is the line that goes around the periphery of the slide. SunQuest is our lab information system, upper right. Epic electronic health record. So we have a very broad network with probably 20,000 IP addresses. And instrumentation is connected to the wide area network. It all funnels through a single data innovation server up in our data center. So this is kind of a point where everything has a common pathway. The results then, after they go through DI, the QC information is parsed out and sent to the Bio-Rad Unity server, also up in our data center. So we're constantly, 24 hours a day, high volume, aggregating QC results on a Unity server, and it's that data that we use to generate the Sigma statistics. So once this is done, and it's done largely by collaboration between supervisory personnel in the lab, people that understand Sigma analysis, and IT. Once it's set up, it becomes just an ongoing system. If you do it for a single lab, you can scale it up to many labs. So this is our current configuration. There are 10 labs there. Will soon be 15 labs. It all runs off the same network using the same DI and the Unity real-time quality control. So we did our surveys of Sigma, and first pass, this is what we got. Labs, at the time we had 9 labs across the top. These are 2 controls, 2 Bio-Rad controls for alkaline phosphatase. And you see those Sigmas are pretty good. They're above 6, well above 6. So I would say that's a safe zone as far as generating 3.1S rules. And so we did convert that particular test, those 2 levels of controls, to 1,3S. Glucose, maybe 6-ish, not so good, kind of an intermediate. If you look across our whole system, this is just 1 month of data. Yeah, there's 3s, 4s, 11s. So again, a single method is not a single Sigma. It really depends on the dynamics of that testing. So it's not a once and done exercise. It needs to be monitored. Potassium is a very good Sigma method. Nothing really there, well, a few that are 5.3, but that's pretty much, as you'll see, or as we saw, when you monitor this monthly, that's a 6 Sigma method. And so that also qualified for wider open Westgard rules. Sodium is one that across our whole system, was quite low. So this was kind of eye-opening. Sodium is not one of the real strong methods, largely because total allowable error is a lot more narrow than a lot of the other methods. Total allowable error for the enzymes might be 20%, 30%. That affects the sigma, where sodium, as defined by CLIA, has a much tighter TEA. So I think this is a very important thing to do if you have Westgard Advisor. Just survey your methods for sigma, see where you are.